Our paper on breast cancer eQTLs includes a table, this is an excerpt from it, which has information on SNPs that have been identified in breast cancer GWAS, and information about their relationship to expression in Nurses' Health Study FFPE samples. And for some of the tests, we can see that there's a simple relationship, simply tabulated relationship, between SNP and gene. There's a single gene that was identified as a trans, the target of a trans eQTL relationship in an analysis that compares tumor to paired normal adjacent tissue. So for this SNP, the gene is CD70. But for a SNP like 17529111, there are 24 associations identified in ER-negative tumor tissue, and it's somewhat cumbersome to tabulate these, and so to help readers interact with this type of data, we've created an interactive utility called bceBrowser. And that's available in a bioconductor package. So I'll just bring up the package here, and we will start the bceBrowser as soon as that loads in. bceBrowser. Oh, don't spell it incorrectly. And it takes a second for it to initialize. There's no internet connectivity. This is all running on the laptop. And we'll blow it up a little bit. And this is what happens when you start the bceBrowser utility. Why do we need a video about it? Well, it's possible to use it in ways that are not completely straightforward, so I thought I would demonstrate it visually and interactively. When you start it up, it comes up with one of the basic findings. This is a finding in paired in normal adjacent tissue, and it involves the relationship between SNP 614367 and the gene TNNC1. And the first type of visualization we have here is a swarm plot. This uses the bswarm algorithm to indicate how individual samples have expression value related to genotype. That is a very simple representation. For paired tumor normal samples, we have a slightly different swarm representation. We'll look at that in a moment. But this very clearly shows exactly what's going on. In fact, we are dealing here with covariate corrected expression values, as you can read about in the paper. So the mean is going to be around 0, and that's the bswarm representation. We also have this thing called a quad plot, which shows the relationship between these genotypes and expression, in this case of TNNC1, for all 4 different classes of tissue type that we have. So even though the finding is only in adjacent normal tissue, we also let you know what the distributions look like for ER-negative and ER-positive tumor tissue. Fair enough. Now, the next tab is on the statistics, and it turns out that there's only one gene that this particular SNP is associated with, and this tells you more about that gene in the stats tab, the actual log fold change that was estimated, and the p-value and the FDR as computed by Limma. The layout tab tells us just how many individuals there were for each one of the genotypes within each one of the tissue classes, and this is the raw data. Sometimes that will have decimal numbers corresponding to imputed allelic dosage, and then layout R is rounding of allelic dosages when those are present so that we can see the actual genotype classes. So those are the basic 5 tabs. This is the very empirical outlook on the data, and then data stratified. That's often a very useful visualization to get a sense for just what's going on. What is special about the layout of genotype versus expression that makes this a significant finding in the collection of eQTLs that have been reported. Now let's show how we can search the collection by using what's called the selectize feature of this device. Here I can just type in a substring of a SNP that I'm interested in. This one, 11571833, is a coding variant in BRCA2 and it's been found to have lots of associations with different phenotypes and gene expression results. So it's nice to be able to search on this, and in this case you get the ability to search through all of the findings that are related to this particular SNP. And we can pick just one of them here. This was a finding which was significant for ER-negative tissue, and you can see in the quad plot that that does seem to be the case, that those individuals who have two copies or that are homozygous for the major allele have relatively low expression of MAC, but if you have one copy of the minor allele, you have higher expression. The unfortunate thing about this, and I think the important aspect of this visualization tool, is that you can see that this is really just an isolated individual. If we look at the swarm for this result, we can see exactly that that is the case. And this is often the case. The minor allele frequency for 1833 is extremely low in our data set. I don't know whether it's available in the stats here. It doesn't look like I reported that there, but you can see it in the table. We scroll down to the 1833, 1.5% minor allele frequency. So you tend to have just very isolated results for this particular SNP. Let's look at ZNF 570. If we look at the swarm plot, once again we see that this finding is based on a single individual. And you can look at it in context of the other tissue classes that we have. So you can see the same sort of condition holds for the adjacent normal tissue in ER-negative tumor cases. Now let's look at another more interesting SNP. Again, we can just use the selectize feature to search for this 1752-9111, and we see that this has a significant association with EGFR. Once again, we can use the swarm plot to get a feel for what's going on in terms of the number of individuals that are driving this statistical result. In the statistics tab, we can see the whole collection of genes that this SNP has been identified as a potential trans eQTL for, and that's quite a useful thing to be able to search through. I can search for symbols. For example, in this part of the table, I can sort by the p-value. So if I turn this off and want to just sort to find out what perhaps is the least significant finding, that can be done. I'm going to finish up by showing how we can search for and visualize a certain type of analysis here. So we've used the tag pair to identify those eQTL results that are based on tumor versus normal comparison. And so if we look at the swarm for this one, we can see a slightly different representation where the tumor sample values are denoted in red, and the normal samples are denoted in black. And once again, you can see that some of these findings seem to be driven by relatively rare events. So this is a case where we've asserted statistically that in the normal adjacent tissue, there's essentially no relationship between let's say, a median expression of FGB and genotype. On the other hand, there is some tendency to see an increasing spread, let's say, as we move to two copies of the major allele for 3757318. But in ER-negative tissue, there's a very strong negative association with the number of copies of the major allele. Again, you can come back to the data to see that that's driven by very few observations, but the statistics are what they are. And so you can search through all the different results. IL-34 was an interesting one. So there's a SNP that's associated with IL-34. And here there's relatively more balance between the genotypes, and we can see here clearly that there seems to be a positive association between the number of copies of the major allele for 241, and that is moving in the opposite direction when we have adjacent normal tissue. So that brings me to the conclusion of the review of the bceQTL browser. I hope you find it useful.